This method can help answer key questions in biogeochemistry related to denitrification in sediments, which are relevant for understanding the nitrogen cycle and the greenhouse gas emissions. The main advantages of this technique are a low disturbance of the sediment structure and the continuous recording of nitrous oxide accumulation for reliable denitrification rate estimation.
For deep-water bodies, select the sampling point according to the investigation aims. Take note of the position using GPS coordinates and take the measurement depth using a handheld sounder. Deploy a messenger-adapted gravity coring system until the sampling tube is approximately 1 meter from the sediment. Stabilize the sampling equipment for 60 seconds to ensure the correct sediment penetration and recovery of a scarcely disturbed sediment core. Release approximately 1 meter more rope so that the sampling tube penetrates the sediment. Be aware that if the sampling tube penetrates too much, it can disturb the water-sediment interface. Release the messenger while trying to keep tension in the rope so that the corer remains fixed and in a vertical position. Recover the corer by pulling the rope constantly and gently. Once the core is close to the surface but still entirely submerged, place a rubber stopper at the bottom of the sampling tube. Uplift the entire coring system from the water. Inspect the water-sediment interface. It should be clear and not visibly disturbed. Release the sampling tube from the corer and place a PVC